Nu ska vi alltså gå över till nästa del som är Aroseniusfondens stipendium och årets stipendiat. Och den som kommer dela ut detta pris är ju vår kära ordförande Monica Westerberg, så välkommen upp.
Tack så mycket. Detta är ju årets stora glädjehändelse att få dela ut Aroseniusfondens stipendiat. Motiveringen är Aroseniusfondens vetenskapliga anslag för 2019 har tilldelat Iva Pruner, Karolinska institutet, för ansökan. Nu ska vi se. In vitroutvärdering av sekundärhemostasen hos patienter med von Willebrands sjukdom, fibrinklotstruktur och proteinkomposition samt som nya fysiologiska biomarkörer. Syftet med projektet är att undersöka uppbyggnaden av det blodkoagel som bildas i samband med blödningar vid von Willebrands sjukdom. I detta innovativa projekt används för ändamålet ny metodik och målet är att ge bättre diagnostiska och prognostiska metoder för blodstillningsmekanismen vid von Willebrands sjukdom. Projektet skulle kunna bidra till förståelsen av den kliniskt mycket varierade bilden vid denna förhållandevis vanliga blödningsrubbning och därmed också till bättre behandling. Welcome, Iva Pruner. It's my great honor and pleasure to give you this.
Thank you. Thank you very much. Thank you very much for having me today, and it's a great honor to give this lecture. So I understood that you already heard the title in Swedish, so it's a big scientific words. So I will try to take you on the journey to decipher these big words, and I will try to explain what is the thing that we are trying to achieve with this project. But first, I would like to present myself because I'm speaking in English. That's because I'm not from Sweden. I was born in Belgrade in Serbia in 1984, and I finished my undergraduate studies in 2009. I studied molecular biology and physiology at Faculty of Biology, and my graduation thesis, it's more big scientific words, but it is about one gene. It's called TAFI, and it is that gene codes for protein, makes the protein that is involved in process of fibrinolysis. So in 2009, I started my doctoral studies, and again, it was the gene for prothrombin. Sorry. And this protein is involved in coagulation. So I just realized that I graduated 10 years ago on 23rd of September, so I'm also celebrating 10 years in hemostasis research. So I've been working with coagulation and bleeding for 10 years now, my whole career. So after my thesis, I started working as a head of biomedicine department in a research center that is specialized for work with elementary and high school students. So I'm very dedicated to scientific education also. And in 2018, I came here and became the postdoctoral researcher at Karolinska Institute. So my scientific interests are completely in coagulation and in hemostasis. I started my career on this part here. So I was more in the thrombosis part, and I researched why blood clots more in some people. That is cool, and why we have diseases like thrombosis, like venous thromboembolism, like strokes, and what is the role of hemostasis in those diseases. Because I'm a molecular biologist, I like genetics very much, so I dedicated myself to thrombophilia. So I was included in the research of molecular mechanisms in thrombophilia. So I researched how is that trait of clotting more inherited. So what are the risk factors that lead to too much clotting, and how can we predict why some people clot more than the others? After that, I changed sides and I went to the part of bleeding. So now I'm included in the research of hemophilia. So we are interested to see why some people bleed, how can we make them clot more, and are there any other factors in the blood that can contribute to the better clotting? And during that process, I became interested in von Willebrands disease. And it's also a bleeding disorder, and we know that there are several distinguished disease types, and sometimes it's problem to know which type of disease is which. And we wanted to see also if we can use some new parameters from our research for the better diagnosis of this disease. And some of my personal interests, as I already said, are scientific education and science popularization. So I already showed you a lot of big words, big scientific words in the title of my research, but now I would like to try and explain each word separately so we can together see what are the things that we are going to do. So we are going to explore the process of hemostasis, and that is one of the most complex processes because it's not one process, it is not one event. It is a lot of events that occur in the human body. Are we back? Oh, we are back. Okay, thank you. So the main role of hemostasis is to maintain normal blood content and normal blood flow. So we cannot have too much clotting, and we definitely cannot have bleeding. So in the case that some injury happens, it is the hemostasis and all the processes included in the hemostasis that will try to stop bleeding. And of course, I already mentioned the hemostatic balance. It is the purpose of hemostasis to maintain that balance. So we cannot have too much procoagulants and we cannot have too much anticoagulants. We need the balance so our organism can function normally. Hemostasis occurs in several stages. Of course, when some injury occurs. First, there is this is a bit strange, sorry. Here we have injury. So when injury happens, some tissues and molecules that are not usually in the contact with blood are now in the contact with blood. And for our organism, that is the signal that something is wrong. So that needs to be repaired. First, the first thing that will happen is that local tissue, local muscles will try to contract to close the wound as much as possible. But after that, a lot of molecules and a lot of proteins have to be activated so the injury can efficiently be closed. And we call that primary hemostasis, secondary hemostasis, and in the end, it is the fibrinolysis. After that first try to close the wound, we will have some molecules and proteins, the von Willebrands factor, which is the main player and the main subject of this talk. He will come first, actually, because von Willebrands factor will see that there is an injury, there are some molecules that shouldn't be there, and he will attach. That protein will attach, and it will say to all other participants, hey, I'm here. Please come here and close this wound. This is wrong. So that is the primary hemostasis. At the end of primary hemostasis, we will have platelet aggregation. So we will have the first plug at the site of injury that will be formed by the structures present in blood that are called platelets. And after that, von Willebrands factor and all the other participants that are already present here will help to start the events of secondary hemostasis. And that is to make this big thing here. I will try, maybe it's easier here, yes. This big thing here called the fibrin clot. So the fibrin clot is the one that will completely close the wound. In the end, we will have the fibrinolysis, and that means that we will try to retract the fibrin clot because we don't need it anymore. So we will try to destroy it and try to establish normal flow after the injury is healed, normal situation. Okay. So in the events of secondary hemostasis, a lot of things happen. A lot of coagulation factors need to be activated so that we can get a final protein called prothrombin activated to thrombin, and then the thrombin will activate fibrinogen to get the fibrin, which will form the clot. So that kind of clot we call a fibrin clot. I won't bother you with all the activation steps. I can just tell you it is really complicated and really efficient, and most of the blood disorders with clotting and bleeding happen because something is wrong with the proteins here. But of course, the secondary hemostasis is something that we are focused on in this project. So, von Willebrands disease is the most common inherited bleeding disorder, and it affects blood's ability to clot effectively. 1 in 1,000 individuals in the world has the symptoms of von Willebrands disease. The thing that happens in von Willebrands disease is that the patients either do not have enough of von Willebrands factor or they don't have it at all. So that means the process of primary hemostasis is greatly affected. So that means we don't have that first guy that will come and say something is wrong, we need to fix this. And that's why people with this deficiency of von Willebrands factor bleed a lot. This disease affects both males and females, and the disease is divided in several types according to severity of the symptoms. So we have types 1, 2, and 3, and type 2 is further divided in 4 other subtypes. The symptoms of this disease are can be seen in early childhood, and children can perhaps easily bruise. They have prolonged nosebleeds. there is excessive bleeding present after the injuries or surgeries, but in the adulthood, the women are more affected because of the menstrual bleeding and all the complications that can be connected to pregnancy and childbirth. So, how is this disease inherited? It can be inherited, it has to be inherited through parents, and it is divided according to the disease type. So, in most cases, it is inherited dominantly. So that means that only one parent is a carrier of the defected gene, and in that case, that gene can be inherited by the child. The good news is that according to, if we look at the disease type, the symptoms can be really mild, or there can be no symptoms at all. That is a good news. But in some cases, like type 3, type 3 is in type 3 there is complete lack of von Willebrand factor. So the symptoms of the disease are most severe, and it is inherited recessively. That means that both parents must be the carriers of the defected gene. And of course we have all the cases also in between. But the foundation is we have to have affected gene. It can be only one copy of that gene or two copies of the gene, and based on that, the severity of disease is different. And that is the main problem with this disease. Because medical doctors sometimes have problems to diagnose this disease. There is a huge spectrum of symptoms, and they could be really mild, or they can be quite severe. It is a general opinion that type of von Willebrand disease is correlated with severity of bleedings, but there are some exceptions. Type 1 is generally considered as the most mild, but sometimes we can observe severe bleedings in this type. And sometimes there are mild bleedings in type 3, even though we would expect really severe bleedings. And sometimes patients have negligible symptoms that overlap with normal individuals. So it is hard to diagnose it and to determine the right treatment. Because of that, even though this is a monogenetic disease, we have only one gene, von Willebrand factor gene involved, the disease is considered multifactorial. And that multifactorial nature clearly shows that there are undiscovered disease modifiers and some other physiological determinants that should be investigated. And that is where we enter the story. This is our research team that is involved in this research. Jovan Antovic is a research group leader, where I am the postdoctoral researcher, and our collaborator is Dr. Anna Ogren, who works with these patients, and she collected all the samples. And last but not the least is our PhD student, Yanan Zhong, who did a lot of experiments in this research. And our project wants to see what other factors might be involved in pathology of this disease. So we are focused on microparticles and the structure of the fibrin clot. So these are the events of secondary hemostasis, even though the von Willebrand disease is mostly disease of primary hemostasis. We just want to see what happens later. How does the lack of von Willebrand factor affect all the events that come after the first ones? So microparticles are small membrane spheres, really small. They are present in blood normally, and they originate from different cells. We are focused on microparticles that originate from platelets mostly and endothelial cells. Microparticles can be procoagulant, and that is the most important trait for us because we are investigating bleeding disorder. So we want to see if we can use the microparticles to achieve better clotting in patients with von Willebrand disease. These little vesicles are released from the cells when they receive signals to release them. We don't know a lot about the role of microparticles in von Willebrand disease. That is why this is, I would say, one of the first studies that will be done with this subject. And the other part of our research is connected to the fibrin clot. And these are the images from our laboratory. Here you can see how the fibrin clot looks under fluorescent microscope. And these are the microparticles. So we have to research fibrin clots because its structure can fluctuate and change, and it is quite affected in a lot of diseases. So, as I already said, the fibrin clot is the last step of coagulation. It is the structure that we close the injury. So it is quite present in bleeding disorders that these structures have some defects. They are not thick enough, so they're not able to close the wounds. So that's why it is important for us to see how these structures look in patients with von Willebrand disease. And another part of our research is to see if microparticles can be included in the fibrin clots. Can they interact with the fibrin network and making it more thick and capable to close the wounds? So here you can see one of our images. These are the patients with hemophilia. We try to see how these structures look in patients with hemophilia. And the other picture is from the patients with thrombosis, where we detected microparticles attached for fibrin network. So now we want to see how will this look in patients with von Willebrand disease. So our goals are again some big scientific words. It's to define the physiological and proteomic landscape that underlies the complexity of clinical phenotypes that are seen in patients with von Willebrand disease. So what are we actually going to do? We will perform some analysis to see what's going on with all the events of secondary hemostasis. Then we will examine how fibrin clots look in patients with von Willebrand disease, and we will try to see if there are any microparticles bound for fibrin clots. And the other part of this research is to see what actually do we have in that fibrin clot, because it is not only the fibrin or fibrin and microparticles. There is a lot of other proteins that are capable to interact with fibrin network, making it more thick or making it more loose. And we are really interested to see what are those other proteins. So for each of these goals, we have a specific laboratory methods that we will use. For example, flow cytometry will allow us to count the number of microparticles in the samples of the patients. Global hemostasis assays will enable us to see the events of secondary hemostasis. I already showed you the images from microscopy, so this microscopy will enable us to actually see fibrin clots. And we also plan to use some proteomic research, proteomic methods. So proteomics will enable us to break and cut fibrin clots from the patients and actually see which proteins are bound in the fibrin clots. So what would be the significance of this research? First of all, and which I'm most interested in, is basic scientific knowledge. We want to see what is the role of the microparticles in von Willebrand disease, and is there any role at all? Can we use them in any way to improve the bleeding symptoms in these patients? Then we want to see how fibrin clots look in different types of von Willebrand disease. And is there something we can do in that part to make them more efficient and better in these patients? And are there any differences in fibrin clot structure between different patients? So can we use that as a parameter to improve diagnosis of this disease? And of course, in the end, we want to see which proteins can be found in those fibrin clots. And can we use that parameter to differentiate different types of von Willebrand disease? Other goals, I would say, are more convenient for medical doctors, because we would really like to see if any of the proteins that we are planning to research can be used as biomarkers for von Willebrand disease. Of course, if we succeed in finding any of these biomarkers, that will contribute a lot to the personalized therapy of each individual patient. And of course, we are hoping to find new diagnostic tools to ease the determination of disease type. So in the end, I would really like to say thank you to the members of the Jovan Antovic Research Group and all the staff of clinical chemistry at Karolinska University Laboratory. And special thanks to CSL Behring, who gave me the Nordic von Willebrand Scholarship last year, so I was able to start my research. And I would like to thank to Arosenius Foundation in the end for making it possible to continue this research. Thank you very much.
Thank you very much, Iva. Thank you so much. Just a few questions.
Yes.
And also, if you have any questions, please feel free. So do you have any idea today, before this trial, about what kind of proteins would activate or interact with the fibrin clot?
In von Willebrand disease, no, because there is no any research that is done in that area. But we have some idea about other diseases, but it's most thrombotic disorders. So we are in the other part of the hemostasis. The problem with bleeding disorders is that usually fibrin structure is really affected. So they are loose and they are barely forming. And it is the problem to get them efficiently in vitro so you can further analyze them. but we still consider it is important to try and do that, because if we manage to do that in von Willebrand disease, that is not as severe as hemophilia is, we will be able to transfer and translate that research to hemophilia and see what's happening there.
Okay. And would you look into the speed by which the clot is built?
Yes, that is the part with events of secondary hemostasis that we will do. We have several different assays developed in our laboratory which serve specific for that purpose. And I can say that after 15 years of use, we are quite an experts in the secondary hemostasis.
And micro particles could they affect by the speed of building this clot?
Actually, yes, because Jan and Jong, that is present here, is working. Her PhD thesis is about the role of microparticles in hemophilia, and we have some evidence that microparticles help form fibrin clots in the samples with hemophilia A.
Okay. Thank you very much.
Thank you. Yes, of course.
Yeah, could you repeat the question?
Can microparticles be anything? Yes, there is a lot of research with artificial membranes and particles which are used for drug delivery. And yes, they can basically do that. But we wanted to see, our idea is to use the microparticles derived from that same patient to treat that same patient. So that is something we are working on. But you're probably right. If we can make them artificially and make them immunologically inert, we can definitely achieve a lot more than with this approach.
And if there is more micro particles within the clot, would it be harder to dissolve the fibrin?
That is what we don't know, but I hope that we will know in a few years. It can be tricky because they are procoagulant, so they obviously have some role in coagulation, but we still don't know what is the interaction and what is the nature of interaction of microparticles and fibrin clots. So that is something that we need to see. At the moment, the thing that we saw in hemophilia is that they definitely interact with fibrin network. But how they interact, that is something that we are working on at the moment.
Great. Any other questions? Well, thank you again so much, and good luck with your studies.
Thank you very much.